So, good evening, dear participants. Welcome to session 4 of this Genome-wide Association Studies module. And we have Pinka ma'am with us for continuing his session on Genome-wide Association Studies. So, over to you, ma'am.
Good afternoon, everyone. Good evening, everyone. Everyone can hear me?
Yes, ma'am, you are perfectly audible.
Okay. So today we have to uh, trim the variants which yesterday we have identified. As I have told you yesterday that we have identified the SNPs and we have identified it by two methods, the de novo and the reference based identification. So the today's session is on we have to trim, like we have identified thousands and lakhs of SNPs. Now we have to trim them. We have to identify those SNPs or the variants which are true, means which are of high quality and which can be used in our further study. So for the SNP pruning, uh, see the first two ways you can see my screen, I feel. The VCF file which you have generated, I hope everyone has that VCF file. Please check if you have. I have shared with you. So in that file, how you have to start, I will explain here. First, you need a Tassel software. Okay, you go to Tassel website, download Tassel. You write down. Tassel is a very popular software of Buckler's lab.
Excuse me, madam.
Yeah.
So if there is no any phenotype file for the genotype file, so how to create a phenotype file for this experiment?
Whenever an experiment is done for Jivas, it is only performed for a specific phenotype. If you don't have the phenotype, how will the Jivas experiment be complete? You have a trait specific genotyping of the samples you have done. For that same samples, you are phenotyping them. Are you able to understand what I am saying?
Ma'am,
No,
kindly
madam.
repeat, your voice break in between.
Okay.
So, please repeat.
Yeah, I am saying that, see,
Okay.
Jiva's experiment is performed for genome-wide association for identifying the variants which are associated with the trait which you are studying. You want to study, like you want to see a disease and a non-disease patient. Then your phenotype is the disease and non-disease patient. You know that the individual which are being genotyped have their phenotype as disease and non-disease.
Okay.
Right?
Okay. So, but disease or non-disease, there is it will come like a case and control.
Yeah, case and control.
Okay. So further, for this, we is very essential for the phenotype file along with the genotype file to do the GWAS analysis. So sometimes we get the only VCF file from the machine, um, uh, SNP-RA machine or some clinical data. So
Yeah.
how there is nothing. They never give any phenotype file for separately. So how can we manage according to that without any phenotype file, but only VCF file, number of individual case and control, whatever in the single or multiple VCF file. So there is nothing phenotype file. So how to create manually? Is it possible or not?
See what
phenotype.
happens. The thing is that in such case, when you do not have a generally for experiments, there is a separate phenotype file. But if you do not have a phenotype file separate for any clinical data, then you should convert your VCF to a PED or PED and MAP file. And PED file has a column which contains information about phenotypes. So you, in that PED file, you can feed your information that the data, your clinical data is of case or control, disease or non-disease, or it is of high or low quality, means high a specific trait, like it is of zinc content, then it high zinc content, low zinc content. This way, whatever is your trait which you are studying can be separately added if you want in your PED file, if you don't have a separate phenotype file.
Okay. So we need to manually create the phenotype file for that. Am I
In
right?
that you have to see for a normally all the studies I have done till now, we have a phenotype file, separate phenotype file, okay, which has the individual's name and the phenotype value, what it is, either it is control or case, or if it is any quantitative trait, then we will write. If we are studying the length or we are studying the age, then we will be mentioning that age as a phenotype. But if you do not have, then you can add in your, you have to convert your VCF to PED and MAP, and that PED file, there is a column of the phenotype where you can add your phenotype and process it further.
Okay. Okay, madam.
Okay.
Yeah.
Okay. So see, yesterday we have complete, as you all have seen, we have got our VCF file, we have identified our variants, the SNP. Now this next step today is this Buckler's Lab Tassel software, which is a trait analysis by an association, evolution and linkage. This software can be used, will be used by us. So all of you who have Windows or Linux, according to their convenience, kindly download this software from the I will copy this link in your chat so that you can download this.
Hello, ma.
Hanji.
Yeah, please, ma. Sorry, sorry to take you back. I wanted to find out about the de novo map PL. The input files are FASTQ files. The input files for the de novo map PL, they are FASTQ files.
Right. You're right. Please say further. What you mean to say? I didn't got your question. Hello, can you repeat your question?
We ran two analyses yesterday, the ref map PL and the de novo map PL.
Right.
Yes, I was able to run the ref map PL of my own, but the de novo map PL, I'm trying to find out if it's the FASTQ files we are going to use.
FASTQ.gz also it takes. What I have shown you the run, it was running and the output has come. It was FASTQ.gz because our file which I have sent you was in the zip format only, FASTQ.gz.
Thank you.
Okay. So I will proceed further. So whoever is using Windows or Unix, they can download it from here. I have downloaded and installed this software. This is a very popular software which is used for everything, which very popularly used for GBS analysis, GWAS analysis, and for SNP pruning, SNP filtering, SNP for visualization, and also final association can be done using this software. So we will be using this GUI because it is very easy to use also, and by this you will be very clear what we are doing. Okay, so first I will explain you how to prune your SNPs. Okay, that is how to, by pruning we means that we have to remove few SNPs, that is filtering of few SNPs. Okay. So in the tassel, you first this video you can see. First you will go to file. You go to file, open your file, the genotype file, the and the phenotype file. This genotype file is actually, first we take input of the VCF file is converted to HapMap file. I will show you how it is done. You go to tassel, file, open as, you first import your VCF file which you have. Just hold.
Ma'am, your voice lost. Ma'am?
Yeah, you can hear now.
Yeah, ma'am.
Yes. So I was telling you that the VCF file which you have obtained, we will be which I have shared with you, which finally you have got. That VCF file we will taking as input in our Tassel software and converting it into HapMap format. That HapMap will be further used. Okay. So I will be just converting that. Someone has written something. I had a query, ma'am. If there are multiple variations in the genome from a reference, then how can we map this variation maps to this phenotype? See, we separately make a genotype file with the variation compared to the reference genome, and we separately have a phenotype file. Okay, so this is the first file which we have created, VCF, is the genotype file which has the variation, and the phenotype file is normally a separate file which have individual name and the phenotype value. Okay, so this software for association like Tassel, Gapit, all these have this work that they will associate your variants with your phenotype. Okay.
Yeah, it will be great how to prepare genotype, the type of TXT phenotype, separate files. So how to prepare this? If you say the steps, it will be great for us.
How to prepare a phenotype file? You want to know?
Yeah, genotype.
Oh.
You having this three file, PNG file and genotype text file, phenotype text file. How you got that step? If you show, it would be great for us.
You want to know that, see, genotype, I have told you how we got yesterday by running ref map PL and denovo map PL. We got the output file, the genotype VCF file you got.
Yeah.
You understood?
Yeah.
Now you want to know how to make the phenotype file. I will
Yes.
explain you. See, currently we don't need for this session the phenotype file. We will need it tomorrow. So if you want to just now to know it, then I can explain just now, or I will explain you just now. See, like this is the phenotype is simple text file. You will write the name of individual one, and then you will write its phenotype. Like for example, it is case. You will write case individual 2, and this name should be exactly same as that is the genotype file. Like there we have mentioned sample 1, so we will be writing here sample 1 only.
Ma, your screen is not visible, ma.
Okay, I will make it bigger. Now is it visible?
There is some
Ma'am, your
disconnection
screen is not
visible.
so your PowerPoint is PowerPoint is visible.
This is not. I will share again.
Yeah.
Yeah, now is it visible?
Yes, ma'am.
It's
Okay.
visible now.
Yeah, so you write sample 1, then tab, and you write what is the phenotype, or space, whatever you give, and then you write the same sample name as was in your genotype file, and sample 2 case, then sample 3 control, sample 4 control. Okay, this is the information we have shared with. We have this is the phenotype file for the just the 4 sample, because here we are currently this is a short, just a template which you can use as many as samples you have. Like if you have a population of 1000 or 200, you will go on increasing and writing the phenotype value. If you have a quantitative trait, like it is of height of the plant, then it will come to be like 22. If it is in 20 centimeter, then it will be 22. Here it is 24. If it is here, control is there, it is just 20. It depends on what is the phenotype.
Okay. So if we have VCF file contains, for example, 100 individuals with
Yeah,
case and control. So we need
yeah,
to write 100 lines in the phenotype file.
right, exactly.
Okay. Okay. Thank you.
So, madam, so I got how we need to create the phenotype file. So, what I was asking is that so in our VCF file we could have multiple variations, and so are we considering that all those variations are mapped to a particular phenotype, like disease or control? So there might be a different type of case and control. Like one might have diabetes, one might have other disease. So these are two various different phenotype. So,
Right.
so how do we know that this particular variation in the gene maps to this particular phenotype?
See, the thing is that this association analysis, which will be performed in your next class, this is for that only, that initially we have identified sample wise variant in our VCF file. We have identified that how many SNPs are present in sample 1 on each chromosome, that we have identified. That was our yesterday lecture which you have seen. So when that has been identified and we have the phenotype of that individual, then the association test which we will using different model, that MLM, GLM, those will statistically check that whether this trait of this or the trend of the trait of our samples and the SNP, it will statistically identify that SNP which is associated with this trait. That is the thing which we will be doing our tomorrow class, the association study, which is based on linkage disequilibrium. It will identify which SNP is in LD for that trait, and it will give you the locus which we will be able to see in our Manhattan plot, that which is associated with our trait of interest.
Okay, madam, thank you. Got it.
Suppose if we have multiple trait of interest in the phenotype.
Right.
So if we have in the phenotype file only one
Right.
column of the trait. So
Yeah.
multiple trait, if we have to write multiple column for the multiple traits in the phenotype file.
Actually, the option, the very normal thing is that you make separate phenotype file and run separately with the genotype. The genotype is same, you have several phenotype. Okay.
Yeah.
So you have more than one phenotype and give it with the individual genotype. Other way is that you add individual columns to it, and you have to, for that you have to mention it while running, that if that there is a multi-trait data.
Okay. Okay.
Okay, the easiest way is to run that in the separate phenotype files.
Okay, madam.
Did you understand?
Yeah, I understood, madam. Thank you.
Okay. So here we have our file. See, we have this VCF file from our data which we are running. We will take this in Tassel. In Tassel, as you all have installed, you will import open as, format is VCF. Our format is VCF short positions. Okay. And then take that file which is there, where VCF file which is present in my system. I'm just searching it. I have saved it here only. If I don't get, then I will go here. Jiva's training. Yeah, this is the VCF. Okay, now this VCF will get open. It will take a bit time because it is a large file. So once it get open, what next steps we have to do use, I will let you know. See, it is loading in the lower side. You can left side, you can see it is opening. It is shown. Now it is open. So it clearly shows that we have used the VCF is generated with our with the 4 samples, and it is showing that there are 4 taxa. That's why it is saying that it has 4 taxa. Then it says that number, if chromosome wise, if you see how many variants are there, it says that on this chromosome CM02869001, it has 7 these many sites where different variants it has identified. 76,946 on this chromosome, it has these many sites. On this chromosome, it has these many sites. That similarly it is showing for all the contigs present in the, because there were many other than the chromosome, there were some unfinished contigs in the genome also, so it is showing that also. If you want to say, this is the site numbers it has shown. So now what is our aim, and you can see that it is showing the change in the different variants with position wise. This is the position. Here it is a specific position which it is saying it will change if you move the cursor. Okay, now as our aim is pruning, so I have in the video in this I have prepared that you open the, after you open the VCF file, you will you need a genotype and the phenotype. This is the genotype file which is in HapMap format. So we will convert this file to HapMap format. So we will write it file save as HapMap in the same directory where you can save wherever you like, because but just you should know your path. So you will be going to our training folder. This is the folder. Here we will be saving the HapMap. HapMap is a format. It contains the same information in other format. Okay. So it will save your file with this name HapMap in that folder. See, it is converting. It is saving. Once we have the HapMap file, writing HapMap, there is some space issue in the system, so it is not able to write complete file. The file is bigger. So I will explain you. See, once you get the HapMap file from your after converting VCF to HapMap, then what you will do next is that you will take your HapMap file, that is the genotype file and the phenotype file, and after that what you will do, that this is the phenotype as I have explained you, this is the genotype. This is way you import your files. Now if you want to trim that file, what you will be doing, you will be going to see what you will be doing. Open the VCF file. First you will open your VCF file, or the HM. This is the same, sorry, I will show you here only. Actually it is not converting. I will be converting on this only. So see how you can prune it. You can prune it by converting into, there is an option, see, thin sites by position. You can thin the site by position. What is thinning by site by position is that you have to what you will be doing is that you will trim the SNPs in a window. If there are redundant SNPs in a single window of 50 base pairs, you will be trimming it. It will ask the distance, in how much distance you want. It is in 1000 distance, you will trim in a region of 1000, minimum 1000 distance. And the output file name will be, you can create whatever you want. That is the trim. This will be the thin, based on thinning. We are thinning this number of SNPs, that is thin side by position. So I'm naming it thin side by position. And and we have also mentioned how much distance it has thin. So it will create this file, uh, the trimmed file in that folder. Okay, so that will contain lesser number of SNP, and it will remove those SNP which are whose R square is very similar. Means if the correlation is very high in them, then it will trim those SNP. It will thin those SNP. The file will be created there. The next way filtering you can do is filtering based on allele frequency. Filtering based on there is a filtering based on allele frequency, filtering based on quality. So for this, we will be using a software called P-Link. You can download P-Link. How it runs? First, I will tell you from where to download P-Link. You go to P-Link. It is by the Harvard Lab. Here, the option of download. You will download P-Link according to what platform you have. I will give this link. And once you download this, how will you trim your file? I will tell you. From here, once you download it, this is Linux P-Link. The Linux based this file, and whichever file you download according to your system. And once you download it, then the codes which you have to run, I will explain you that in my PPT. See what you have to run to trim your file, your SNP. You will be using P-Link, NoWeb, my file. You will make my file P-Link, and this is the saying, the command was showing you that based on from the beginning I will run independent pairwise, means in window of 50, if the 5 base pairs, if 5 SNPs have correlation more than 5.5, then trim them. So it is mentioned here. If you see, it is mentioned here. Just hold on. It is showing here. So it will display you how it is trimming. See, we have given this command independence pairwise, and your file, it will show that 0 case, 0 control, and then it says the total genotyping rate is this much, and the number of SNP with missing genotype how much, and then it will tell you that how many SNP it is pruning, 2802 to this many. These many SNP it has pruned from specific chromosome. Now you will get, if you now the output you will get with this VCF, several new files, the pruned files. Okay, so the next command we will be using is that just hold. Is it clear to you? I feel it is clear to you all. So now the next is extract prune. In the, like we will make the bed file of it and extract the prune data out of it. So we will be using NoWeb file, my P-Link, extract the P-Link, extract the output of the prune file. So we will be doing it this way. I feel you got this. And the other filter you can use here is my P-Link, and you can use MAF filter, which will give you based on minor allele frequency. It will remove all those variants which have frequency lower than 0.05. Okay, so this way you can do the pruning of your SNPs. Once this is done, we can see the structure of if our individuals are from diverse population, we need to see how is this structure, means how is the diversity among the different individual. This is a very popular software structure. You can download it. I will give you the link. Once you download this structure, install it. I already have it. Yeah. Now it will, this needs the HapMap file as input, and it will show you the structure. So I will take the data as data from structure. So the data set we can use can be of any type. It can be a AFLP data, it can be SNP data. Structure is a very old and highly used software for structure analysis of the population since very long. So structure here, like we can take any of the data which we want, like we are dealing with SNP. So we will be taking SNP and microsatellite data from HDP panel. We can get it here. We can download it and see its structure in the Xstructure software.
So it's good for after SNP pruning.
Yeah.
We can use select this structure software.
See, yes, after you have pruned your SNP, means you have removed all the low quality SNPs and you have removed the redundant which have very similar correlation. Then we go for structure analysis to see, and every data doesn't require structure. It is only required when you want to see the diversity of a population. Like in case control study, there is no requirement of such structure. Okay.
Okay. Which file format is required for the structure input file?
HapMap, which we have just made the HapMap format using Tassel.
Okay.
That is required.
Okay.
I don't know if this site data will get download. Just wait for a while. Somewhat the site is low, but you just have to open the data file, give the number of individual information. Let this get downloaded once, then I will be telling you. I have a HapMap file earlier. Just hold, I will get that HapMap file. So I got the HapMap file. One HapMap file I have, I will, so that I can show you how does it run in the structure. So first you have to open the HapMap file. You can see my screen, I feel.
Ma'am, your screen is visible, but your voice is low.
My voice is low.
Yeah.
Okay. Just hold on. Yeah, is it okay now?
Little bit okay, ma'am.
Yeah. Still it is lesser?
No, no perfect, ma'am. No perfect.
Okay, okay, okay. So we got our file. Now we will import our file in from our training folder. This is the HapMap file, which is after converting it to after converting the VCF to HapMap using Tassel. Okay, it will get opened here. Because the file is bigger, so it is taking bit time. Let's wait, or then I will trim the file. I will mail you, I will send the HapMap file to you all also, so that you can run it separately on the structure.
Yeah, it's very good for if you upload all the training files,
I
genotype
have
and phenotype.
earlier I have shared all the files which I have used except this HapMap.
Okay.
This file is too big in size. That's why this system is unable to open it also.
So
So
there is one
I'll
file,
be,
thin site by position. Can we upload that as well?
yeah, you can upload it. That is a smaller file than that. I have taken lesser sample. We will uploading that only now. And now I will rerun the structure so that because it got hanged with that bigger file, the bigger sized file I had. So I will be using open data file that by thin by site. Let's see. Actually, this today I'm using a Windows system that has lesser capacity than my earlier Linux system. Might be this because all work I did on Linux, but today I was using the window interface as in window installation of all these is easier. So I wanted to explain you from there because everything can be done on terminal, but the thing is it takes more installation time. So I feel this is not able to take the data of thin also. I will try one more file. Let us see it takes or not. HapMap. This size is also bigger. HapMap.hmp. Let's see if it takes. Yeah, it has taken. See, the file which we uploaded it says that it has 153,959 lines and it has 15 columns in it. Okay. Now we have opened. Now we'll be making a new project. In the project, you have to first write the name. We'll write the test name of the project. We will browse it. We'll give it the path where we have kept our all files. And choose data file. Here also we can directly use the data file. Earlier I just was opening just to check whether the file is getting opened or not. So we will be using the data file. So it is asking that in your data file, how many individuals are there? You have to mention it. How many individuals are there? How many, what is the ploidy level? What are the number of locus and what is the missing value? So you can ask it for the show the data format. So it says that your data has uh, 1 line, 4 columns and 132180 lines and 15 columns. So as we know that we have 4 individuals, and the number of loci we have were, let me check the tassel file. It will tell us the number of loci it had. 4 individual and 162,7. This is the number 162,75554. Okay, you have to mention it there. 27554, we will mention it there. 162,75554. And missing data we will be is present in -1 format. Okay, so next, the rows are markers are in rows, and the data file stores data for individual in a single lane. Rows, there are marker, and in a single line the data is, this is a special format we will not be giving, we just give this information. Next, it will ask the individual ID has been mentioned. We'll say yes, it is mentioned. The phenotype information we have not mentioned, because we will give a separate phenotype file, so we have not mentioned it. So we'll be finishing it. So it says that test, finally it is telling you that for individual, these many loci, this is the ploidy, this is the missing value, and it will proceed. You will proceed. Okay. Okay, there is one higher number of lines, so it says the bad data entry. So I will again convert our VCF file to HapMap so that it takes. That's why I was running on their test data set, because my data set which we are using is a shorter, is just for 4 sample. The actual data set is always a higher, but the website of structure is not running today. So I will share this link to you. You all can download this data from here, and if you have any problem in running it, please let me know. I feel it will be working after some time, because in the morning it was working. So from here you will download the data from here and then run it in the structure and see how the diversity analysis it looks like. I will explain you the results of its result. How does it shows the result? So about structure, we can study what does the software actually does. Okay. See, structure is like the result we will be getting is like these images, which will show the diversity of your samples. So if we go to the manual of the structure, it will explain us better. Just hold on. We will go to the manual. Picture lab, it is same. Yeah, this is the recent version of structure is fast structure, which is used for very high, if you have very high number of SNPs. Okay, so structures of software, I want to make you understand few important points which are very important in its output, that I will explain you. See what columns it had. We have the label. Each row has a label, which is a string integer, and a population data. What type of population data it is? The locus data, where it is located, the phenotype data, and the extra column, and the genotype data obviously will be there in the HapMap file. These are all the input which it is saying. And missing format is missing genotype is generally mentioned in -9 by convention. Okay. And the output, I will show you how it will give you the output, which is still we have to run in our system. And there are two versions of structure. The one is front end. See here, we started here. One is front end. This is the front end structure software which we downloaded. There is a back end software also in which we do not see the front end. So once we import our data in it, that is what we did. We imported it, selected the directory and the data where it is present, and after that we used the we got the it wanted the information, the window, we got the window which wanted the information of the individual. The what is a ploidy, that is either it is a diploid data or a triploid or a polyploid data. Then what are the number of loci and how you have mentioned the missing data value that you have given. And once you give this information, uh, is completely correct. We will work with the test data only tomorrow. First we will work with this, so that because this data, if your machine is not very robust, then if you have more than 100 or 200 individuals, it will give some, it will not work. So here we will, they have like used a smaller data set, so we will be using a smaller data set for this. Once the data set is loaded, see it is showing, as it is mentioned, these many labels. This is the population ID and these are the locus information which got opened. Once this, after this step, what comes, that it asked how many, what is the length of burn length, how many times you want the this diversity analysis iterations to be done. So it asks for the burn length and number of replicates after the burn you want, so that you only give a smaller length in the beginning, smaller period, and then you can further modify it to get a better result. Because if your system has less robust, that it might get hanged. So once this is done, then what it gives as the output, this is running. Okay, these displays will be coming to you, and now it has given the select the simulation to run. How many, it asks what, how many clusters you expect in your, how many k value, it represents the clusters in your population. So this is a very general, the default value that we gives. One cannot be the cluster is only diversity is only studied if it has more than two types of population in it. So minimum is 2, and then maximum here range they have given 5 because the population is small, just of 150 individuals. And then the number of iteration, how many times you want to repeat that k value which you are setting, that and output, how many times you have to repeat it. Once you set this parameter, it will give you a result like this. So this result shows you, this plot shows you that your population data set is lying in, has three type of population, the mixed population with of three types, one blue shown by one other red and one by green. So if you see by in a triangular, in a dot plot type view, you will see this, these were four population, but the groups they are forming is three. Means two population are merging, somewhere yellow population is merging with the other. So that's why the only three groups are being formed here. And this, to in order to identify the correct k value, next step is that we will be running the structure harvester software, which will be telling us that we have, like here we obtain three. You can see that it shows 3 as a k value, and 3 was the 3 were the number of populations shown here. And more popular is this figure is of structure. So but it has, we will run structure, the structure harvester is run by people to know, to assure that how good is your k value, means which k value, because if you give 3k, it will give you result at 2k also, and because we have given 2k, 3k, 4k, so but the best result it got at 3k. So it, you can use structure harvester to revalidate your structure output. So that is also a very popular tool which is used in diversity analysis. And like the trimming which we did today with peeling and tassel, that can also be done with VCF tools, the trimming part. Similarly, so because actually for doing a thing there are many, not just one tool, there are many tools. So VCF and VCF tools and BCF tools are also the tool by which you can do trimming. If you have a very large data set, sometime running structure is really tough. It takes like my data set which I am currently, I wanted to show you, it took me around more than 1 month to run on my machine, because if you give higher number of iterations, at times it takes a lot of time, because it is a very it is a very complex burn iterations process. So that's why it takes a lot of time. It calculates many values and then gives you the results. That's why always, if you just want to learn it or practice it, if you don't have actual data, please run on a very small data set. Best is to run it with the test data set given by them, so that is the optimum for any machine to run on. Okay.
Madam,
Hanji.
I didn't get training file. Please share with me. Share the files.
Okay,
Yeah.
I have mailed the encode group. I feel they can mail you again, because I don't have your direct email ID.
Okay.
So I feel they will if you write a drop a message, they will mail you again. I have shared all the training data set.
Ma'am, I have already uploaded on the day one all the material in the drive link.
So is
No.
that link active now?
Yes, ma'am. The link will be active for next 6 to 7 months.
Okay,
But the
okay.
training file we didn't get. Other file is there.
Training file contains
The VCF file,
all the files.
genotype file, and HapMap file, like that.
Okay. You are asking for HapMap file. HapMap file today we have prepared. That I will be sending you, and the phenotype file also I will be sending today.
Yeah, including VCF file also.
VCF file is there. You see,
Yep.
I have opened that VCF file only.
Okay.
This is the SNP name. This is, see, it is mentioned, it is a VCF file. Okay.
Yeah.
So, but now
Yeah.
for the analysis, we will be working with HapMap. So I will be sending you the HapMap, genotype, and the phenotype file.
Okay.
Because VCF is itself a bigger file, and this VCF which we have generated here is for the lesser number of samples, which is not the actual data set for 4 samples. If you have, it will change when you have the actual population of 200, 300, or 1000 samples which you are using.
Okay.
Okay.
Yeah.
You got my point?
Yeah, I got it.
Okay.
And then in the structure,
Yeah.
I saw the summary of the picture.
Yeah.
There is a sort Q and K value.
Yeah.
What is the K means? I'm not understanding.
K is basically, K value is defined for the clusters in the population. Like you want to know that your individuals are how diverse, how many cluster of populations they are making, the group of population, the diversity. If there are many groups made, that means the population is too diverse. Here
Okay.
it is showing in, it is falling in 3 clusters, means the population individuals are having genotype range which is in the, they are clustering into the 3 different clusters only, they are not very diverse.
Okay.
So it is clustering based on the genotype data of your genotype data.
Okay. So based on the K value, how to predict the which sample is more diversity or less diversity? How to predict from the picture?
See, if your key value, what you like your population, if you expect your population to be very diverse, and if you're getting your k value of after running structure to be higher, then you can expect that your what you were expecting, your hypothesis is correct, that it is a diverse population, because higher the k value, more clusters it will be formed,
Okay, it means more diversity.
more diversity.
Okay.
Ma'am, how this K value has impact on the market rate association now you further go forward?
See, this is a market. This is we are not doing any association here. Here we are. This is just the genotype file. We have not taken any phenotype file as an input here.
For Jivas, we are using structure analysis.
Yeah, this is an diversity analysis which is done before running association analysis. If we want to know how diverse the population which we are going to use for association analysis, so people do diversity analysis. This is an additional analysis before running the association analysis.
Yes, ma'am. So how this diversity has, how do you ensure that your population is ideal population for this market rate?
Like if your hypothesis is like you feel that your population is too diverse, so it will always obviously contain many different types of marker, like it will have not just one type of marker in it. So if our aim is to identify variants to associate or related to a trait in a very diverse population, so structure software helps us to find that whether our population is really diverse or not.
According to you, how much would be the optimum population size for Jiva studies?
Optimum at least above 200, not optimum, at least it should be above 200 samples or the individual, but if it is as much as higher it is, then the better will be the GWAS result. Because if you see, like I have a paper in type 2 diabetes in which we have more than 10,000 individuals.
Yeah. So human genome is a big genome, but some plant species will be having lesser genome. Just I wanted to know.
Yeah, in plant generally people, I see that they make very smaller population also, 200, 300.
K value I'm asking, what would be the optimum K value?
What K value they take? They take generally, I've seen that in papers, they go with 3K value.
Okay.
In plant. Are you working on plants?
Yes, ma'am. Yes.
Okay, which plant?
Carrot, actually. I'm working with carrot. We have
Carrot.
done.
Okay.
We had the 3K K value 3.
Yeah.
Whether it is optimum.
Madam, excuse me, madam.
Yeah.
So suppose if we have 100 sample, so any one of the sample it shows very lowest diversity in the structure. So we can we keep the lowest diversity sample, or we have to eliminate?
See, lowest diversity will not a single sample. Lowest diversity should be present if we have lesser number of we have lesser groups of samples. It is a group value. It is not a single value. Like if you have a single sample which is showing in a single group, that is very rare, because here input sample is a population data. We have taken many sample from a single population.
Okay.
And the sample which is very different from all others must be having very different SNPs, and it must be getting removed also.
Okay.
Ma'am, I have one query. Hello.
Yes, please say.
I'm having some context I have generated from RNA-seq data. I have some context. I want to know I want to identify SNPs between two samples, SNPs and indels. Which tool you suggest, ma'am?
For RNA-seq data, see, ideally RNA-seq data, if it doesn't have high number of sample, then identifying SNP, I don't know what is the aim of your study, but identifying SNP from RNA-seq data with just 2 sample, I don't feel is.
These 2 samples are highly contrasting samples and geographically different samples actually.
Okay, geographically they are different. Yes, then you can identify. You can use the software we have discussed, that is you can use stack, or if possible the most easy is you use BCF tools, because your sample size is so small. So because stacks take information of population, so it will expect the two different population data set. So better to use BCF tools.
No, I have some 1,000 genes only. Out of 40,000 genes, I'm extracting some 1,000 genes which are differentially expressed. So only for those contexts, I want to know the
If you only want to just two sample and only 1,000 contig, then it is just like you are you can align those two samples files, and this you can see find the changes at the same position. There is no need to run any calling software.
I need also. I wanted a list also for each gene. I wanted a list. Which is the most
Yeah, see, the thing is that it is a very simple, like when you align it both with the reference in a normal MSA alignment also, you will be able to see, but that will be a manual work. So with the MSA file, the MSA output, if you get it in a written in a text format, then you can see at which position what is present.
MSC means what?
MSA, I mean multiple sequence alignment.
MSAL, okay.
Yeah, or pairwise alignment also, because you just have two sample. But if the contigs are very high, you can very easily use BCF tools. It will be very easy for you.
These contexts were obtained from de novo assembly. So further, I have to map them back to the samples. That's why I'm.
Yeah, I understood your point, because see, it's a de novo. See, de novo based variant calling is not very popular, is not very common. So that those contigs, if you want to use a reference based method, then use those contigs as a reference.
Okay.
And if you want that no, you want a de novo based, then you can use that de novo based software, which will take your samples as to prepare the contig reference based contig, that basically with the sample file, your raw FASTA file, it will prepare the reference genome. De novo based assembly, what is done,
Okay.
it will reassemble your contigs basically.
Thank you.
Okay.
Yes, ma'am.
So I feel everyone is clear.
Ma'am,
Yes, please say.
yesterday I repeated all the analysis and I'm getting error at the last step where we run refmap.pl command.
Okay,
And
what
I
is,
always got the error that it cannot unable to open the BAM file.
but
So
why
can
is
you
it so sure? Why is it because people in this group have run it? There must be some specific issue. What is your error?
error is failed to open BAM file.
That
SAM
means
file
the
only.
location
SAM,
you have not mentioned the correct.
no,
Like,
binary file
I'm in
cannot
the
to open
same
in the
directory.
text file.
Binary file cannot open it. You have to give its path. Why to open it? You have to give its path in
I am
refmap
saying
PL.
that I give the path and the error I got is that fail to open your BAM file.
That means your path is not correct. Then only it is not able to open that file. I have shown you running, and students on this group have run also. You have the BAM file with you. You have generated the BAM file.
Yeah, I use that BAM file even in the same directory, but still I'm getting same error. I don't know.
Your path, the path which you have given in the command must be 100% not correct. Otherwise, this error will not come if you have the file, because the dash dash sample folder gives the dash dash sample option is for that only. You can share the exact command you are running, so that I can once see, and the path of your folder.
Ma'am, we can give a liberty to share her screen, so she can share her screen if possible.
Yeah,
Yes,
you can
ma'am, I can share.
So
you can share your screen.
share your screen. I have given liberty to share your screen.
Yes, show me.
Ma'am, here is my command which I run. You can see. And here where I'm getting error. Can you see my screen?
Where is your pop map file?
Yeah, it's here.
It is in here. MNA's analysis. Okay.
No.
Keep the
Here,
sorted file and pop map file at the same your BAM file and the pop map file in the same location.
Actually, I pasted both. Is also here with the BAM file. It is also here.
Yeah. So which location you have given? Jiva's analysis is the folder. Jiva's Linux analysis. Show me where there what you have mentioned. Yeah, you have mentioned refmap t. Here you have not given the name of your pop map file. See, here it should be the name of the pop map file in pop map. First, this is wrong, and after.
Map input.txt.
But here, see, your command starts here. Jiva's reference. This first line it starts here, and then it goes this. Then you write output here, and this sample is. Why are you writing this sorted.bam? Don't write this sorted.bam. You have to give the path of your file. You don't have to give the file name. Just remove that sorted.bam.
So I can run it again.
Yeah, don't use that. You do this, it will run. Okay.
Ma'am, please wait 1 second. I can.
Your voice is not coming to me. What you are saying?
I'm saying please wait 1 minute so I can run. You can see my error, please. I just copied it and pasted here. And you can see it's still giving the same error.
Why is it giving to you? 4 files, 2 population and 1 group. Error failed to open. mtnfp. Is this path correct? Can you type this path mtnf python? This path which you have given, because I didn't find mtnf python session 17. Jiva's analysis. Okay, mtn 5 star. And this Python 2.7 you are basically running it at the in a conda. Your conda is activated, I feel, right?
Yes, Conda. This is the name
Anji?
of my Conda. PY2.
Hello?
So kindly respond to ma'am.
Hello?
Yes, ma'am, I'm hearing you. I'm saying this PY 2.7 is the name of my Conda environment.
Yeah, just once deactivate the conda. We have not installed this stack on conda.
Actually, I installed with Conda. Okay, I can.
Okay, I got your error. So you deactivate it, and the problem is that your the input, the name you have given is sample1 sample1. You have what you have mentioned in your pop map file. Show me your pop map file.
Also sample 1.
You open it.
Okay.
Yeah, it is sample1 parent sample7, and your sorted BAM file are in. Just close you this. Your sorted BAM file are here in the sorted BAM folder. You want to give sorted BAM or direct BAM?
I tried both.
What if you want just sample1 BAM? Then it is okay. Then it should take sample1 parent sample7 BAM is there. Then it should take. But if you want to give sorted BAM, then you give the path till sorted BAM, and in your pop map file add that add the exact name of the file present in sorted BAM.
ma'am, I understand what you said.
Okay.
And yeah,
Your voice has again gone. Hello?
can you hear me now?
Hello?
Yeah, I'm saying, ma'am, I also tried sorted BAM file and even without BAM file. But I don't know why I'm getting this
Okay,
same
you
error.
again come that has your Conda been deactivated?
So I can repeat it
No,
again.
you have to deactivate Conda. The command is not this. Just don't write py 2.7 once.
Okay. Conda deactivate.
Yeah, and now run
Okay.
the command.
So I can give the same path to my simple BAM file. So,
So you please do this, and then you can send me a. We can discuss in the next class. Okay.
ma'am, can you please share
Okay.
your email? So if I get problem, I can
No,
share.
you send on the group VR. Okay.
So you just write to me. I will share it to the ma'am. Okay.
Okay. Thank
Okay.
you.
Thanks for today's
So
session,
thank
ma'am.
you, everyone.
And
Yeah.
we will meet again with ma'am tomorrow with his with her last session on genome-wide association study.
Yeah.
So tomorrow will be the
Yes.
last session of ma'am. So hope you people are enjoy learning with the good life. And we thanks ma'am for sharing her experiences on genome-wide association studies. Now I seek permission of ma'am for ending today's session.
Okay.
So thanks.